So, if you want to understand what is common between Wnt signaling and Hedgehog signaling, what are the points that are common between them? Very good. So, when not needed meaning when the pathway is off, when the pathway is not active, right? That's what those two pathways talk about. No, when the pathway is off, the key transcription factors which are supposed to function, which are the ones who do the job, they are degraded, right? So those are the two pathways that we saw, and both of them they are morphogen kind of, no, which there is a gradient of the secreted ligands which go outside, and then there is a gradient which happens, and that's how it will give rise to some of the other signaling pathway. Anyways, so both of those pathways, what was core or what is at the core for both of them was ubiquitination. So let's take a step back and see what ubiquitination is, because it will become a little bit more relevant in the pathway that we are going to talk today, so that you guys will have an idea about it. So as most of you guys know, ubiquitin is 76 amino acid polypeptide, right? There is nothing new that we are going to add to this. So this person in 1975, Goldstein, he found that it is a protein which is ubiquitous. That's how ubiquitin name came from, because it is everywhere. And this is what it is shown. This is the amino acid sequence for ubiquitin, and the lysines are highlighted over here. We will go to those lysines in a bit. And so it is covalently attached to the target protein, and it is covalent modification, but it is reversible, meaning a battery of enzymes can go and covalently link ubiquitin to the target protein, and there are set of enzymes that they can remove ubiquitin from the target protein. That's how there is a balance between ubiquitination and deubiquitination happens. So ubiquitin, which is the 76 amino acid polypeptide, is encoded by 4 different genes: UBA52, RPS27, UBB and UBC. So essentially we are not going to go details into how it is happening and all that, but just remember that ubiquitin is encoded by 4 different genes. It is highly conserved throughout the eukaryotic evolution, means it's present everywhere, whether you take plants, lower order eukaryotes, higher order eukaryotes, anything and everything, it is there. And of course it does lot of functions, right from regulation of proteolysis, which is the protein degradation or protein turnover, nuclear localization, the differential localization of proteins is regulated by ubiquitination, and we will understand a bit more based on the different ubiquitin chains, the different functions are there. Then chromatin structure, which I think I might have mentioned about H2B, the ubiquitination. So all these histone tails, they get modified, right? All the acetylation, methylation and all that. So there is one example of H2B is there, histone variant H2. Then there is H2B which gets ubiquitinated on a particular lysine, that is lysine 120 if I am not wrong, and that is very important for transcription and initiation, and it regulates the expression of subset of genes like that. Then genetic integrity, of course it is very important for DNA damage responses. There are certain proteins that would get ubiquitinated. Protein quality control and signaling. Protein quality control, you guys must have heard about ERAD, no? Have you guys heard about ERAD? ER associated degradation, you guys know. So let's say we have protein which is getting misfolded in ER. Most proteins undergo folding, right? So if there is a protein which undergoes misfolding, or there is it's beyond repair, then it has to be removed out of this one. So who does it? That there is ubiquitination dependent pathway which takes place, where there is P97 which is AAA ATPase, retrotranslocation will happen, P97 pulls it out of the membrane ER and then takes it to the proteasome, which is another barrel like machinery which is full of proteases and all that, that will chew off the proteins. That is your ERAD, ER associated degradation. So, again ubiquitination pathway is multi-step process. There is E1, E2, E3. There are 2 UBE1s present in humans. There are 50 E2s which are ubiconjugating enzymes, and E3s are the ubiquitin ligases. Over 600 enzymes are present. So, how ubiquitin gets charged? You have E1 which essentially takes up the ubiquitin at the expense of an ATP. Then these E1 will transfer ubiquitin onto E2, and then E2 will associate with your E3 which is ubiquitin ligase. And then ubiquitin ligases, there are multiple mechanisms. Either this ubiquitin ligase will take the ubiquitin on itself and then it transfer it to the substrate, or it associates with E2 and directly it will transfer from E2 to the substrate. So, this is the general mechanism of ubiquitination. Now, you can say ubiquitination also has, like whenever we talk about this histone code, no, people call about histone code. Have you heard about histone code or epigenetic code any time? Disha, he is sleepy. Epigenetic code, have you heard about? Yes? No? No one? Yes, yeah. So, essentially histones get modified, that is what we talked about, right. Let us say acetylation or the methylation, which are the most common type of post translational modification which happens on your histone tails. So, it is called as code, meaning there are some enzymatic machinery which is going to go and modify these histones, that they are called as writers, because they will add certain those molecules or those groups, like there are histone acetyl transferases that will go and acetylate something, or DNA methylases, those will go and methylate the DNA. Those are writers, they are going to add some information to that, that is why they are called as writers. Then there are readers, meaning there will be some set of proteins which can go and read or recognize the modified form of histones. They are called as readers, they have the ability to read. Like you guys remember, we talked about SH2 domain containing proteins, right. So, if there is a kinase which goes and phosphorylates, or there is a receptor which gets autotransphosphorylated, these SH2 domain containing proteins can be called as the readers, because they can go and bind to these phosphorylated tyrosine residues. That is how it. Similarly, we have so many other enzymes, other molecules which are called as readers, because they can go and associate, read what information is there on those histones, like that. And then there are erasers, meaning they will come and remove those marks, like hats. So, hats are histone, what is not hats, what is called? HDACs, histone deacetylases, or there is DNA methylation removing enzymes are also there. What are they called as? Some TET enzymes are there, like they oppose DNMT1, like that they remove those marks. Similarly, in case of ubiquitination also, there is this process, readers, writers, readers and erasers will be there. Of course, writers, as I mentioned, there is ubiquitin ligases are the ones, they will essentially add ubiquitin to the target protein, and here you can see that this is slightly not very clear, but there are 3 types of ubiquitin ligases: Heck type, ring type and ring between ring. So, RBR is ring between ring. So, these ones, if you see over here, these are ring type, meaning these proteins or these ubiquitin ligases, which are proteins, they have some specific domains. The one of that domain is called as ring domain. So, that is their catalytic domain. What they do, they have the ability and they have the substrate recognition domain onto their N terminus, where they go and bind to the substrate, C terminus will go and bind to E2, and this E2 will be carrying the ubiquitin, and then they transfer ubiquitin from E2 to the substrate molecule. And if you guys remember, this ubiquitin is always added onto the lysine residue on a target protein, or there is M1 linked, which is the first methionine can be added, but mostly it goes through lysine residue, right. So, there is ubiquitin has last amino acid is glycine. So, there is this glycine lysine linkage happens. Then there is second type of ubiquitin ligases, which are called as HEC type ubiquitin ligases. These HEC type ubiquitin ligases have the ability where they can bind to the substrate, they associate with E2, they take ubiquitin onto themselves first and then they add it to the target molecule. So, the difference between ring type and HEC type is that ring type directly transfer ubiquitin from the E2 to the substrate. HEC type they take it on themselves and then they add it to the substrate. And these different type of mechanisms, how these ubiquitin ligases add ubiquitin to different proteins, it becomes very important for the substrate specificity as well as to have very specific, because that is when the specificity comes. It is not a random process, there is a specific ubiquitin ligase. So, there is a different protein will be recognized by different ubiquitin ligase when the combination of E2 and E3 becomes very important, because these are the ones which are going to decide what kind of polyubiquitin chains will be formed. For example, there is a ubiquitin ligase called NAD4, and then there is ubiquitin ligase called E6AP. Okay, these are E3 ubiquitin ligases, both are of them HEC type. I am just giving you additional information, no need to remember, even if you miss this it is okay, but just understand how the combination of E2 and E3 will form different ubiquitin chains. E2, so there are I said there are around 50 E2s, right. One such E2 is called as UBCH5. So, when you have UBCH5 and NAD4 ligand, these two guys protein together, which gets secreted out and it has certain roles to play, it undergoes some kind of modification, some post translational modification together, they will form UBK48 chains. So, the combination of E2 and E3 becomes very important to form the kind of ubiquitin linkages they want to form. So, and so this is too tiny to read for you guys, but these are the readers, meaning like you had SH2 domain containing proteins which could go and read the phosphorylated residues, tyrosine, phosphorylated tyrosine molecules. Like that, there are so many proteins which have the ability to go and recognize ubiquitin or ubiquitinated proteins, and these proteins they have different types of different domains like UBA domain, or there is VHS, or there is GAT, and then there are so many of those ones, the whole list is there. So, this is not necessary that you know and remember all of them, just understand that like you had a molecule, a certain protein with a specialized domain which could go and recognize and bind to the phosphorylated residues. Similarly, in ubiquitination also they have the ability to go and bind to ubiquitinated proteins through the ubiquitin binding domains. UBD is nothing but ubiquitin binding domains, and there are several of them. In fact, some of these ubiquitin binding domains will again have specificity. They can bind to K63, K48, monoubiquitin and what not, different based on the biological context. Is it fine? Are you guys following what I see? Then there are deubiquitinases, which are erasers, nothing but they will remove the ubiquitin molecule from the substrate which are ubiquitinated, and there are two types. There are around 100 deubiquitinases which are also called as what is it called as metalloproteases, because they are dependent on some of the other metal ion. So, for example, there are two types of deubiquitinases which are cysteine proteases and metalloproteases. Okay. Cysteine proteases means they use they have cysteine as their catalytic residue, which is important for the deubiquitination, and metalloproteases, they dependent on some metal ion. For example, there is a deubiquitinase called AMSH. It needs zinc ions. It needs zinc ions. Similarly, we have different types of deubiquitinases, and if you guys go and start digging out the literature, we will really find that there are very specific, like this AMSH which I said, it has been very well studied deubiquitinase, which can go and remove only UBK63 linked ubiquitin chains and no other chain types. So that makes them very special and very specific, so that they can do the specific job in the cell. Is it clear, Durgesh? Are you still following? Yes? No? Yes? Good. Tanvi, did you understand? Good. Now, there is something wrong here. This one I got it from a paper, but I realized that this is not correct. The topology of these chains should be very different than what they have shown. The linear ubiquitin, so something like this. You see here, K48 has this kind of topology, K63 has slightly different topology. Topology meaning how the ubiquitins are arranged in the polyubiquitin chain, whereas this cell research paper, मैंने शेंडी लगाया उसमें कुछ तो लपड़ा किया. So and they have tried to give the importance of the different ubiquitin type of polyubics or different types of chains and where do they function. So most of the membrane proteins which are supposed to undergo degradation or wherever, they undergo K63 linked polyubiquitination, whereas the soluble proteins, like for example any protein, Rutuja, right, any soluble protein which is supposed to get degraded because its lifetime is over in the cell, they will undergo proteasomal degradation, but they are tagged with K48 linked polyubiquitin chains. Then whenever there is a DNA, there have been other examples, like there are monoubiquitination when the single monoubiquitin is added. This NF-kappa B signaling, whatever they have shown, that we are going to talk about it in a while. And then when there are some other types of DNA damage response related signaling, when it happens, there is lysine 6 ubiquitination happens. So like there are 7 lysines. So here K6, K11, K27, K33, what else is there? Here lysine K6, K11, K27, K29, K33, K48, K63 and methionine. No, ubiquitin is a polypeptide, it's 76 amino acid polypeptide, it looks like this.
So it gets attached to the substrate?
Yes, onto the lysine. It gets covalently linked to,
This attaches to the amino acid?
yes, lysine. So if you have a protein and this protein is supposed, let's say this protein's life is over, we need to somehow get rid of this protein from the system. There has to be a mechanism how the machinery will understand कि इसको उठाना है. How do we do that? Ubiquitin serves as a tag where there is a specific machinery which will go and recognize this protein, like your HEC-T3 ligases. Then they will attach this ubiquitin on a particular lysine residue on this. Once it gets ubiquitinated, it's kind of a flag that इसको उठाओ. Then the machinery can come, then there are certain readers, they will go read this ubiquitinated protein and take it to the proteasome. Proteasome, it can get degraded. So it's a 76, it's like phosphorylation where your ATP is used, the gamma phosphate of ATP is transferred by kinase onto the serine, threonine or tyrosine residue. Similarly, ubiquitination happens. There are similar modifications like ubiquitination, like sumoylation. That's another, sumo is a small polypeptide, right? It's like ubiquitin that is covalently linked onto the target protein. So these kind of modifications do happen. Now, after telling all this about ubiquitin, maybe we should have discussed, as I said, we should have talked about it maybe before we started talking about your wind signaling or hedgehog signaling, but there it was ECPC where we talked about K48 link, but now you guys understand little bit more about what ubiquitination is. Yeah.
Last slide. So what is the main? So the first the linkage type. So let's suppose in the first one lysine 56 or lysine 6.
Hmm.
At the 6th position of lysine, this ubiquitin enzyme are being attached to this lysine protein, amino acid. So that is called what parking is called. So what is downstream, that is USP 30? What is
No, they have given example of deubiquitinase. So what they have done over here, I'll explain. So let's say you have a protein. Okay, here there is a lysine residue. What happens? You have ubiquitin. This ubiquitin itself has different lysine residues. Okay, this K6, K11, K30, 20, K whatever, K27, 29. These all are lysines which are present on ubiquitin, not the target protein. So target protein, there is a lysine. This ubiquitin has a glycine. You see this glycine over here, double G, RGG, this is the glycine. So what happens? Your HECT E3 ubiquitin ligase first will come and attach, there is this one, so this lysine will be attached, there is a covalent modification will happen. So essentially this ubiquitin is attached onto the lysine of your target substrate protein. This lysine can be anything on the protein. That also there is a way to determine which lysine it is, but we are not going in detail. So let's understand this is a protein which needs to get ubiquitinated. There is a lysine and then there is ubiquitin which are. So if you only one ubiquitin comes and attaches, this becomes monoubiquitination. Why monoubiquitination? Single ubiquitin came and attached, right? So if there is another lysine here, another ubiquitin will come and attach. Only single ubiquitination. In this case, now what happened? Here there is multi-monoubiquitination, meaning there is more than one single ubiquitin added. So it becomes multi-monoubiquitination. Now what happens? These ubiquitins which are present over here, those ubiquitin itself has so many lysine residues which can be modified. So the next ubiquitin molecule which is gonna come, that ubiquitin will be attached onto any of these lysine residues. So if that ubiquitin comes and attaches onto the lysine 6 like this, then it will become lysine 6th linked ubiquitination. So it will form a chain of ubiquitin, 1, 2, 3, 4, 5, 6, 7, whatever ubiquitin which will be added, that every time next ubiquitin ka lysine 6, next ubiquitin ka lysine 6, like that it will keep adding, then it will form K6 linked polyubiquitin chain. Similarly, if it is K48, it will form K48 linked polyubiquitin chains. That is how the polyubiquitination will happen. But every time a new ubiquitin will come, it will come and bind onto the existing ubiquitin, the lysine any of the lysines on the existing ubiquitin. Is it clear now? That is how. So they have given example. Let's say
this attachment has to be some limited number, or else like it will come and attach, it will come and attach. There has to be some limitation of this attachment.
I think they can form. So what happens is a continuous process. Now what happens? Let's say first ubiquitin bind, second ubiquitin bind, third ubiquitin, fourth ubiquitin. So, it forms a tetraubiquitin chain, or there is hexaubiquitin chains, right. Once these chains are formed, there are readers which are surveilling around, right. They will go and bind to that. Once it binds, then it will do whatever it's supposed to do. Sometimes these chains can be very long chains as well, sometimes they can be very short chain as well. So, I think if a protein gets K48 linked polyubiquitin linked K48 linked to ubiquitinated, after tetraubiquitination or something, these proteins can be moved out, they can be taken to the proteasome. There are certain ubiquitin readers, they recognize tetraubiquitin means 4 ubiquitin chains. Like that it could happen. So, there can be longer chains, I do not know how long maximum chains people have found in the cells, but what I know is that there are hexa and more than that chains also do happen.
This chain arrangement has to be done to form a structure.
Yeah, once the ubiquitin keeps binding onto that, it will the topology of those ubiquitin chains will be very important, so that the readers can go and recognize them. So, K48 will have different topology, that is what I was trying to tell you right here. You see here K48, it looks like this. Like this. So, in real structure there is a structure to that. What I am showing is this example. If you have K63, it starts looking like this, like this. Even linear ubiquitin and K63 has similar topology. So, the readers can recognize between them. So, the way these ubiquitins are added, the chain of ubiquitin topology will determine sometimes which protein will come and bind to them. And the example what they have given is like K lysine 6, mostly E3 ubiquitin like parkin. Parkin is an ubiquitin. So, have you guys heard about mitophagy any time? No? Sudha? Mitophagy, you know autophagy? What is autophagy? Cell eats itself. Anybody else want to tell anything to that? Most of the, if there is a bad cell, a bad organelle, or something which is not good for the cell will try to get over with that, right? Then you take it to the lysosomes and degrade it. This process in general, there is macroautophagy, microautophagy, xenophagy, and there are 1,000 different types which are coming today. I mean, it's kind of killing system, but if you ask Harshita, they will think that it's a survival mechanism, but it's a different way of looking at it. But it's a very interesting story. The person who discovered autophagy, he was, what is his name? I forgot. A Japanese scientist. It starts with O. Yeah. So when he started, yeah, when he started working on these autophagy genes, those are called as ATG genes, he used to work on yeast. So he used to delete one gene and see what is happening and all. So in his interview, he was telling, he was not bothered about what's gonna come out of this. उसका क्या ट्रांसलेशन होने वाला है, क्या प्रोडक्ट निकलने वाला है, कौन सा स्टार्टअप बनने वाला है. He was not bothered कर मुझे नोबल मिलेगा, nothing. He had a small lab, he used to check the phenotype, and he never even knew that something will come out of this. And then you see today he won Nobel because he discovered those ATG genes and their functions. So basic scientist does have value, which people don't appreciate today, but I was talking to, when I was a PhD student, I was talking to one of the Japanese scientists, and I asked him, like, I was still finishing my PhD. I think I finished, I was about to defend. I said, I want to go to the next stage in my career. What is the suggestion you would have for somebody like me? He said, choose a problem which not everybody is interested in solving it. Second thing is, but you should have the courage to pursue, to address that problem till the end of the world. That's how Japanese people are. You go and see, they don't try to do everything that whole world is doing, no? They come up with their own issues, they will try to follow their own path. So, I kind of was impressed. He was very old Japanese scientist. That's what they do. This example of autophagy person is that. Okay. So, autophagy, going back to your, so like mitophagy, what is mitophagy? If you have bad mitochondria, you get rid of them, right? Xenophagy is something, if you have anything which is foreign to the cell, if it enters, you get rid of them through this mechanism. So, mitophagy, this parkin is ubiquitin ligase, that becomes very important. It goes and ubiquitinous the outer mitochondrial proteins which are bad. That is how the parkin is another important, and this enzyme has very important role. I think some of the neurodegenerative diseases also are associated with it, and they have given examples of all other different. So, linear ubiquitin, there is only one Lubac karke, there is a system which can add the only linear ubiquitin chains. So, yeah, if you guys must have heard about APC, anaphase promoting complex, that also has that associates with different E2s, and then it will form different type of ubiquitin chains. So, it's very interesting. It's very huge space, and people have done a lot of work into that, and still it is very interesting. Anyways, so coming to this, now let's talk about NF kappa B signaling pathway. The reason I gave introduction to ubiquitin, as I said, we should have discussed earlier, but better late than never. So, we will learn something more. Now, we will learn about why it is more important in case of NF kappa B pathway. So, so you guys know about NF kappa B, what do we know? Anyone? No, have you heard about this? Gaurav? Very good. Gaurav, have you heard about this pathway any time? Never? No problem. Anybody else? Shayan? Inflammatory cytokines. Anybody else? Oh, he knows the mechanism. Vaishnav, have you heard about this? Name, good start. No problem. So, as you said, this is mostly, it was actually initially discovered as because it led to the transcriptional activation of genes which encode the light chains of antibodies. You know, antibodies have light chains, heavy chains. Most of you must have been asked this question in your PhD interviews. How many chains are there on antibodies? Shritoma is like, nobody asked me this question. No? इसको पूछा है. Was this asked to you? No. I usually ask this because there are two chains. Now, how do you prove that there are two chains in antibody? So, if you take this antibody and run on SDS page, how many bands do you see? So, these kind of questions we ask initially, right? So, there is. Uh-huh. Huh? Huh? No, there are two. No, one is light chain, one is heavy chain. That's what we get. But the next question is, how do you prove that there are two light chains and there are two heavy chains? Think about it. But this pathway was initially discovered like that. So, now after over a period of years of research and all, people think that it's a master regulator of immune system in mammals. So, interestingly, this pathway has been studied in flies as well. Even though flies do not have immune system, they don't make they do not make antibodies like what we make, but still whenever there is any bacterial or any other infection, they make this what is that called as? They make these antimicrobial peptides. That's how this pathway was discovered in flies. So, flies do have very important these things. So, NF kappa B pathway, it is known that it indicates the NF kappa regulated systems that have been conserved. So, essentially it is conserved across from the flies to the higher order eukaryotes or mammals, where in most of the cases it does the immune cell functions, where it is very important in whatever called as the immunogenic responses. So, of course, it gets activated in immune cells whenever there is bacterial or viral infection or inflammation or when you have the acute exposure to ionizing radiations, which are very stressful conditions and whatnot. So, immune system, when components of bacterial fungal cell walls bind to certain toll-like receptors on the cell surface, again this pathway gets activated, and this get activated by inflammatory cytokines such as TNF alpha, IL-1, and which are released by nearby cells in response to infection. So, essentially it is very straightforward pathway which is activated whenever there is any challenge in terms of bacterial, viral, inflammatory and all other stuff. So, this is the pathway which is not very complicated, but we will go through this one by one. So, if you guys remember when we talked about Wnt and Hedgehog signaling, in Wnt we already knew that beta catenin is the key molecule, there is whole machinery which is trying to come together and degrade that molecule when pathway is inactive, right. In this case it is like it is ulta. Here when pathway is inactive there is nothing degradation happening, but when the pathway gets activated the degradation happens. This is the major difference between Wnt, Hedgehog and NF-kappa B. Wnt, Hedgehog, both the cases when pathway was inactive, the key transcriptional molecules were kept down regulated or degraded, whereas in this case the pathway only get activated, only upon activation of pathway the degradation is happening. किसका degradation हो रहा है that we will see now, but that is the major difference between NF-kappa B versus the other pathways. Now, the key molecule over here is this IKB alpha, or this is the kinase, this is the molecule, this is the transcription factor which is made up of this P65 and P50. Again you can understand when somebody discovered these proteins they ran at 60 kD and 50 kD, that is why they named them 65 and 50, P50 and P65, and that is your this name, what is that, IKB alpha is the transcription molecule. So, whenever transcription factor, so whenever pathway is inactive this transcription factor is sequestered in the cytosol by these two, by this molecule which is called as IKB alpha. That is very important name, remember that. Now, what happens whenever any of these things happen, whether it is viral infection, ionizing radiation or activation of these IL receptors and what not, or there is activation of toll-like receptors, anything and everything that can happen, it will go and activate this complex. So, there is NEMO, IKK beta, IKK alpha, this is the kinase machinery that will go and activate this beta subunit of these this kinase complex where there is NEMO, alpha and beta, this IKK is there. So, whenever there is upon the activation of the pathway by any of these means will lead to the phosphorylation of the beta subunit. Whenever this beta subunit gets phosphorylated, it will go and phosphorylate the IKB alpha molecule which is sequestering the transcription factors P65, P50. Once it gets phosphorylated, of course, ubiquitin ligase will come, it will ubiquitinate this, and only this molecule which is getting phosphorylated will get K48 linked polyubiquitination will happen and it will get degraded. That is your IKB alpha, which is the scaffolding protein which was holding your P50 and P65 molecules. Whereas, and then once they get free, the transactivation domain as becomes accessible, the nuclear localization signal is free, and then through importin mediated transport they will get to the nucleus, and then it will go and do the job that it will go and it stimulates the transcription of over 150 different genes which include your adhesion proteins, inflammatory cytokines, chemokines and what not, all these things. So, we remember the most important molecule is transcription factor is P65, P50, which is sequestered in the cytosol when pathway is inactive by this protein called IKB alpha, and whenever pathway gets activated, IKK beta subunit of this kinase which is associated with NEMO and alpha will go and phosphorylate this. the phosphorylated IKB alpha molecule will become substrate for ubiquitin ligase. Same thing what happened with your beta catenin, same thing what happened with your glia transcription factor. In every case they first get phosphorylated, the phosphorylated substrate becomes a good phosphorylated protein becomes a substrate for ubiquitin ligase, gets ubiquitinated. In this case they are very specially specifying the ubiquitin linkage type, that is K48. Whenever it gets K48 linked, it goes to the proteasome and get degraded, so that your P65 and P50 are free, so that they can enter the nucleus and drive the expression of the genes which are supposed to be activated. Is it clear? Now, the question is, these receptors which is your TNF alpha, IL-1 and what not, they do not have any kinase activity, right? Then how will it go and activate the IKK beta, or how will it go and activate the IKK phosphorylate IKK beta? Question समझा? तेजा नहीं समझा? You did not understand the question? What do we say that in case of this NF kappa B pathway, the pathway gets activated, let us say by IL-1, whenever IL-1 gets activated, the pathway can get activated, right? The pathway getting activated meaning your IKB alpha getting phosphorylated by IKK beta, but this receptor does not have any kinase or catalytic activity. Then how will it phosphorylate the kinase, or how will it activate the kinase? IKK beta. Zoya?
So according to the diagram, this viral infection or ionization radiation leads to the phosphorylation.
Oh, you think that the ionization radiation directly?
I think this receptor would activate the intrinsic kinase activity of IKK.
Okay.
Or maybe to couple itself with some other kinase.
Some other molecule.
Couple itself with some other kinase and then make that kinase.
Good. So here is the answer to that question. So IL-1 receptor, whenever it gets activated, what happens? There is a cascade of molecules which happens. This MyD88, IRAK and TRAP6. These are the molecules which will go and bind to them. Then these TRAP6 and ubiquitin ligase, what happened? This These ubiquitin ligase will form specialized ubiquitin chains which are K63. You guys, you have to remember this pathway has two types of chains coming into picture, K63, K48. K48 are important for the degradation of IKB alpha. In this case, this TRAP6 ubiquitin ligase is building these K63 chains onto your IL receptor, this IL-1 receptor itself, right Muskan? And when you build these ubiquitin chains, that is when your NEMO, IKK beta, IKK alpha will come and bind. The NEMO has the ability to go and bind to K63 chains. This is one of the readers of K63 chains. The interesting part, there is a kinase which is recruited, that is called as this TAK1, and this kinase belongs to the family of transforming growth factor beta activated kinase 1, or it is MAP kinase family, or MAP kinase 37 is this another name. So what these ubiquitin chains are doing here, K63, they are acting as a scaffold to bring together a substrate and an enzyme so that they come to close proximity. It's very interesting, right Shivam? Because your IL-1 get activated, then this MyD88 and the other IRAK molecules, they go and associate, then your ubiquitin ligase called TRAP6 is going there. That is when it will start building these ubiquitin chains which are very specifically K63 type ubiquitin chains. When you have these chains built, these chains are acting as a scaffold, right Durgesh? These chains are acting as a scaffold so that two key molecules of the pathway can come together. Who is getting phosphorylated, Vaishnav? IKK beta first, and what does it do? It goes and phosphorylates the next molecule. Who is going to phosphorylate IKK beta? The kinase called TAK1. So these ubiquitin chains so Nemo has the ability to go and bind to them. So Nemo goes and brings them together. It phosphorylates this guy, gets activated, go and phosphorylate the other guy, and then then this is what happens. Yes.
Sir, this NYDA2, this is also found in case of TLR signaling. So, how does, how this recognizes the receptor, whether it has to bind to this serine kinase or TLR?
No, it doesn't have any kinase.
How it recognizes which receptor to bind?
So I think these molecules, whenever you have this TLR or any inflammatory signaling happening, these molecules can go and associate with those receptors. They are kind of, I think, the relay, or they are the, what you can say, the secondary, the proteins which can go and recognize this activation, the state of, like in case of GPCRs, this beta arrestin 2 goes and binds, no, maybe something similar it is doing. It just go and understand, it just go and recognize the state of activation of this molecule, and then it will recruit the downstream molecules. It's kind of a scaffold. So I don't know whether it has very specific function, maybe it's very generic function to do. And then, because unless and until you do not recruit TRAP6, the downstream chains will not happen, and NF kappa B pathway is going to activate it across, whether it is TLR signaling, IL beta, viral infection. So this pathway will be common to all of them. That's what I understand.
But this chain is specifically for IL-1 receptor?
No. These chains will be there, as I said, even if it is TLR, similar K63 chains will happen, because Nemo can go and recognize them then. So if you remove Nemo, none of the pathway will get activated. I mean, it is little bit more complicated. There is a role of the Lubac also in this pathway, but they have not explained. I don't want to get it more complicated, but it's very straightforward. Did you understand what is happening, right? It's just that the the pathway is very straightforward, something similar like what we have been talking about. It's just the two types of ubiquitin chains are becoming very prominent, one acting as a scaffold and one acting as a degradation signal.
So, generally K63 acts as a scaffold.
Usually K63 chains have been shown to be very important for the trafficking purpose. Meaning, you guys remember EGFR trafficking and all we talked when it comes to the early endosomes, or GPCRs come to the early endosomes. From early endosomes they are sorted into the late endosomes and to the lysosomes for degradation. So these trafficking processes, most of the membrane proteins undergo K63 linked polyubiquitin chains. So this is another example where it is acting as a scaffold. So based on the biological context, this can happen. In general, these K63 type ubiquitin chains are important for sorting and trafficking function. So here also it's doing slightly different function. It's just building the scaffold. And this is the only function which I also know whether K63 chains are important doing this kind of function. I don't remember any other example where K63 are acting as a scaffold.
Also, the length of polyubiquitinization
Is big, because that's how you want to assemble the whole cascade, no?
seems very big. So, how do they manage the length? In certain cases, like K48 polyubiquitinization, the length should be like this tetra.
There might be some literature on the length of the chain which I have not really read about, like how do we determine it, because I think what happens is that, you know, when the surveillance machinery is going on, if you get the enough amount of ubiquitin that can be recognized by the reader, that molecule will be taken out. So in these, specifically in these cases, when you want to build it as a scaffold, I guess, then you build longer chains, and then because not any other and every other molecule which functions or has the ability to go and recognize these chains will not function here. Maybe there is specific context, only those proteins like Nemo who has the ability, and that also maybe Nemo also might be recognizing the K63 chains when they are of certain length. That's how it would happen.
So can you once again explain what are readers?
Readers are the molecules which So, ubiquitin readers, you know, they are the molecules which have which they have the ability to go and recognize ubiquitin, as simple as that. Like your SH2 molecule, SH2 domain had ability to go and recognize SH2 domain, the phosphotyrosine molecules. Like that, these people have the ability to go and recognize ubiquitin. If you take this UBA is one of the ubiquitin binding domain. There are different proteins which have different types of ubiquitin binding domains.
So this UBA act like a reader.
UBA, so any protein which has UBA can go and recognize ubiquitin.
So these readers are itself responsible like for making the change.
No, readers will only read the ubiquitin. Writers will write the ubiquitin. Those writers are nothing but your UBE3 and E2 ligase combination. Right? So, for example, you take this example of this VHS and GAT domains, right? So, these VHS and GAT domains, they have the ability to go and recognize. You see, it's written type of ubiquitin binding. It says mono ubiquitin, means they can go and recognize a single ubiquitin.
Can it only recognize or it also like sends signals that you have to attach at particular site?
It doesn't know who has to attach.
Like if they like these readers itself they recognize a particular ubiquitin. So they signal that ubiquitin to go and attach to particular.
No. So, ubiquitin, this is what happens. You have protein, you have protein which gets ubiquitinated, right? This is recognized by the machinery, E3 ligase, it will come and attach this ubiquitin to this. Once this ubiquitin is attached, what happens is there is a reader which will come and recognize this. Ubiquitin is a flag, it will recognize this. Once it recognizes, based on this reader, they will determine what happens next. If this reader is something which is going to take this protein, if this is K48 linked, this reader will have a specific role, it will take it to the proteasome and then it will get degraded. If this reader wants, like, you know, if these things that you should just change the localization of this protein from cytosol to the nucleus, it will take it to the nucleus and leave it there.
I am just confused that these readers are itself present on the ubiquitin or it is present on the substrate.
Readers are another proteins. So, you have protein A which is ubiquitinated. You have protein B. This protein, so when you have protein A which has a lysine residue somewhere accessible, E2 and E3 will come together and then they will add ubiquitin onto this. So, it becomes your ubiquitinated protein A. Now, what do you want to do with this ubiquitinated protein A will be determined by the next protein which comes and bind to this. So, the protein, that's when your protein B might come which has the ability to recognize this ubiquitin, which is your reader. They are present in cytoplasm. Then what will happen? B will come and bind to the ubiquitin. Then it will decide what to do. If this B, if let us say this A protein is bad protein, okay, which means that system wants to get rid of it. Then what will happen? This B protein is the protein which will take it to the proteasome. So, for degradation, right? If you think that this protein A is good protein, it is getting ubiquitinated in this case only upon signal, then protein B might not take it to the proteasome. Then based on, again, this depends, this lysine, which kind of ubiquitination has happened. In this case it will be 48. In this case some other kind of ubiquitination will happen. If it is K63, not B but C might come and bind to this protein, this ubiquitin. So based on that it changes. Do you have questions, Sanjit? No? Why? He's like, is it clear? Sure, good. Anything else do we have? So we saw three pathways where the intermediates of the key molecules are regulated via ubiquitination, UB Hedgehog, Wind and NF kappa B. NF kappa is different. NF kappa B is different than Hedgehog and Wind because there the key molecules get degraded when pathway is inactive. Here the regulatory molecules get degraded when pathway is activated. Ubiquitination is important when pathway is activated. Ubiquitination is important when pathway is inactive. Is it clear?
So can we say ubiquitination also play role in phosphorylation?
Ubiquitination does not play a role in phosphorylation. There have been examples where the ubiquitination and phosphorylation regulate each other. So, for example, histone H2B. So, there is histone H2B which gets ubiquitinated and lysine 120 residue, and there is a tyrosine which is present next to that, which is tyrosine 121. So, what happens is that whenever it is phosphorylated, it cannot get ubiquitinated. So, there are so many examples you will find like this, where these two post-translational modifications can regulate each other, but that is again dependent on biological context. It's not necessary that it has to happen, but there are some, it just adds additional layer of regulation. I mean, that's it.